very much, and thank you for the invitation. I'm going to split this talk into three parts, and I'll maybe say something about who I am as I kick off. So I'm currently the head of school of the School of Biological Sciences at the University of Aberdeen, and I'm a soil scientist. And in fact, really putting my research and my teaching in perspective, I love soil. I love soil because fundamentally, soil feeds the world. And we can think about vegetation, but also I love soil because it hosts livestock as well. And we forget that in this part of the world, livestock is such an important component of livelihood. I love soil also because fundamentally it hosts the plants that fix the carbon that COP26 is all about. And when we think about carbon sequestration, here we are in Glen Tanner with some beautiful old Scots pine upon some really fantastic sandy pod soils. And then I also love soil because it is the boundary between aridity and sustainable agriculture. And I've done a lot of work in the Middle East, and again, we can see that soil with very, very little adjustment can suddenly feed people, stabilize land, and be quite different from other component parts of the environment. But I spend a lot of my time enjoying teaching about soils, and here I am a few years ago in Iceland, looking at some beautiful deep volcanic soils, 2 meters in depth, and a large international group, and discussing how the soil forms and how it behaves in the wider environment. But perhaps most importantly, I love soil because it's a very beautiful feature, and it's something we don't often see. And we can look at this image and see the colors. We can see the presence of organic carbon. We can see leaching. We can see iron. We can see highly oxidized environments. Soil, regardless of where you are in the world, is a really beautiful feature, and it's part of our environment. But tonight, I'm not going to focus on beautiful soils. I'm going to focus on the opposite. I'm going to talk about soils that we've wrecked through the release of pollution into the environment. And I can think about pollution in several ways. I can think about elemental pollution. Let's look at the periodic table, pollution caused by agriculture and industry. I'm going to touch on hydrocarbons as well, industrial and spills. And also you can think about pesticides, when we purposely apply a poison into the environment because it might degrade or it might stay there in the future. And then finally, and I'm not going to talk about this at all tonight, but it's something maybe to discuss in the future, is the impact of nanoparticles in the environment, because ultimately these will be sequestered into the soil environment. Well, let's start with potentially toxic elements. I use that term because I'm not just talking about metals, I'm also talking about semi-metals, antimony, and arsenic in particular. Now, what we can see here is that there's a group of elements coded red that are what we tend to call heavy metals: chromium, nickel, zinc, and also we can see arsenic and antimony underneath the nitrogen and phosphorus. Now, some elements respond to my first graph on the right of the screen. These are elements that are essential to life, and when they're very low in concentration, we call them trace elements. But as the concentration increases from trace, it becomes optimal. And then once it exceeds optimal, it will become toxic and then perhaps lethal. This includes copper, nickel, zinc, chromium. It includes a lot of elements where they have important health-giving features to all sorts of life. But the second graph shows a different set of elements that includes mercury, lead, cadmium, antimony, and arsenic. And these are elements that we don't think have a credible biological beneficial purpose. And therefore, at lower concentrations, organisms can tolerate them, but then again become toxic, and at some point become lethal. So one of the problems we have within soil is not just measuring the concentration of a given element, but placing it in the context of the harm that it might cause. The hydrocarbons are slightly different, and this is just an example of two areas where I've captured leaking hydrocarbons. Again, in addition to my university position, for a long time I had an environmental consultancy and diagnostic company that specifically focused on cleaning up contaminated soils. I'll use many of the examples this evening. When we look at hydrocarbons, we have to chemically quantify the amount of the hydrocarbon, and that's why I want to use the term fractionation. So I take a sample, I use a solvent to remove the hydrocarbon, and then I run it through a gas chromatograph. And the gas chromatograph allows me to see the different lines of molecules separated by temperature. So as I look at the right-hand side of this graph, I'm seeing molecules that are generally of a heavier molecular weight than I'm looking at the left of the graph. So it allows a separation, and I can then look at those individual peaks and relate them to molecules. Now, what we then think about is how can we relate a measured concentration in the field to the potential harm that could cause to different receptors. By receptors, I mean humans, I mean ecology, and I also mean water. And believe it or not, the key receptor that we use to protect soil is a toddler. Now, what we don't do is give thousands of toddlers soil and then see when there's an impact. What we do instead is we develop lab-based systems that understand the amount of a given contaminant that can be desorbed from the soil and assimilated into the toddler. We choose toddlers because they're small, a small body mass, and they're also prone to licking their fingers and actually taking in more soil than adults. Now, we can actually then correlate quite clearly that there's a relationship between the amount of soil and, believe it or not, the size fraction of soil and the harm it does to a human receptor. And that's widely published. When I think about ecological aspects of soil, I stand back and I see an incredibly complicated relationship. And this shows at the base of this figure here, I've got organic matter, dead waste. What happens is microorganisms will process that and they'll liberate things like nitrogen, which can be transformed from organic to inorganic, and then that's taken up by plants. So there's lots of different functional activities taking place in plants, and we can begin to see how we are impacted by the presence of contaminants. In fact, what I specialize in is looking at biological tests to understand the harm caused by pollution. So I use a lot of different microbial tests, such as measuring activity, the size of it, looking at key processes like nitrification, or looking at enzymes in the soil. I then also use earthworms, because earthworms are easy to count, easy to grow. They live in the soil and they eat the soil and they exude material. You can look at how much harm contaminants do by looking at the impact on earthworms. And of course, I look at plants, because plants ultimately use the soil, they make available different components, and we can look at germination or mature plants in terms of that process. So when I look at ecology, I'm trying to understand the functional relationship of these different components. Now, when I think about humans, I'm able to therefore sit down in the case of organic molecules and relate observed human effects to different concentrations. So if I look at this table here, I'll just pick on some key details. The first thing is I can look at the different fractions, so aliphatic, so straight chains, and aromatic being ring-based structures. I've not named individual molecules here. What I've done instead is I've focused on different fractions. So the first fraction of aromatic, C5 to C7, carbon 5, carbon 7, that includes benzene. And we know benzene is a carcinogen, and it's not particularly good to play about with benzene, to inhale it or to touch it. So we can see here that the residential without plant risk value is 0.613. That means if the value is less than 0.6 mg per kilogram, no long-term harm is likely to be caused by children playing in that material. Now, if I scan down and I look at, for example, or look up C35 to 44 aliphatic, so that's the chains with carbon of 35 to 44, so big molecules. Basically, the risk here is at 27,600 mg per kilogram. That becomes a problem for the toddler. So we can see that we've got one at 0.6 and one at 27,500 mg per kilogram. So great differences. So a lot of my time is spent developing these tables, using data that is validated to try and develop these, and then use this to develop models. And there's a range of models that we use within my group. This here is a UK-wide model called Contaminated Land Environmental Assessment. This is the sentinel model that the agencies use, and it allows a Monte Carlo simulation or to derive site-specific criteria based upon really the values I've shown. And that's freely available and is useful to look at accordingly. But so I can define a cleanup value, I can define a concentration that no longer poses particular harm, but commercially I need to create the link of how can I develop a technique to validate I've met that concentration. And that's what I call predicting cleanup. So over the years, I've done a lot of work on hydrocarbons, both in the lab and the field. And what I developed with my team was a set of simple experiments that characterized the likelihood of degradation taking place in the soil. And it was very simple. It was measuring the indigenous activity. It was measuring the amount of the genes that were induced to degrade hydrocarbons. And then it was about the total concentration of the hydrocarbon and its form, and then also the number of degraders. So quite a simple process. What that enabled me to do is establish a traffic light system. So if the factor of degradation was in the green, it meant it was happening. It was an absolute no-brainer. Degradation would take place. If it was in the red, well, it wasn't going to happen very quickly for hydrocarbons, and it probably required a lot of help, nutrients or augmentation or something else. And then in the amber area was material that was likely to work, but required a bit more of a prod. So commercially, I began to look at sites in orange and try and make them turn green. And that was really quite an interesting way of promoting science. Well, let me give you some examples. This is lab-based work. And this is, I'm just going to call them soil B. These are soils from refineries. And this first one, the experiment ran for 18 months. So we set up an experiment by taking the soil from the field, and then we looked at different treatments. So the treatment was no additions. The second one was a nitrogen and phosphorus addition, an optimal ratio. And then the third one was, in addition to N and P, we also added degraders. Now, what we can actually see is, to be honest, and statistically, they don't make a big difference. So ironically, at the start, there was a speed up, but over time it wasn't. Now, in terms of my end point, so the H residential, that's for toddlers who would eat the soil in the back garden. Eco is for my earthworms and other assays I showed. And then water is the amount of oil left in the soil matrix that, if leached, would be harmful for drinking water. So soil B, after a period of 2 months, but it flattens, it passes the toddler assay, but it fails in eco and in water. If we look at a second soil here, same treatments, very different type of hydrocarbon. This is much heavier, takes much longer to degrade. Even by month 16, this had failed in the residential, it failed in ecological, but it passed in the water. So different soils, different behavior, different breakdowns, different endpoints. But at that time, I was really interested in thinking, well, here's a kilogram in the lab. What happens if we tried 10 tons in the field? So in this case, we went down to the Royal Dockyard at Rosyth. We collected lorry loads of soil. We brought it back up to Peterhead. We mixed it, homogenized it, and then we grew up hundreds of litres of degraders. And then we focused, instead of just a jar, what we did was use bobcats and we turned the soil over on a very frequent basis. So we could actually think how frequently we turned it would change the behavior, because it's both biological and it's physical chemical. Again, there's the windrow being turned within a contained area. Again, I'm adding lots of water, mitigating it, I'm keeping it aerated, but I'm also making it very moist. And then I'm sampling it with colleagues on a frequent basis as well. Right, so when we look at a pile, so this is not turned, this simply is a static mass, very much the same as we saw in the field, in the lab. And here you can see I kick off with a concentration of just 2,800 mg per kilogram. I then see that the control sample stays about the same concentration, but I see a significant degradation in the others. So the nutrients plus inoculum. And then by day 91, I can see it's very much flattened. But what's interesting is when I look at the risk assessment criteria, I see I pass ecological, but I fail both human and water. So what's interesting here is I'm degrading hydrocarbons, but I'm not improving it for human processes. So it's almost like bad remediation, if you like. When I then look at a more aggressive turning process, I can see a much greater reduction in hydrocarbon. In fact, the control sits at about 1,500 mg per kilogram after 10 weeks, whereas all the other treatments have declined much more quickly. And in particular, I can see that nutrients and inoculum reach a threshold in 10 days. Now that's incredible. If I do nothing, I have to wait 80 days. If I'm aggressively managing it, I can do that in less than 10 days. So again, there's a cost benefit if I'm trying to remediate. The problem I have here is I pass residential and ecological. I just still don't pass water criteria in this case. I've still got contaminants that are unacceptable. Now, when I look at metals, it's quite a different story because I can't biodegrade metals. What happens is metals fix into the soil. So I've got two different soils here. I've got very sandy, low pH soil at the top, and I've got a much higher pH, finer soil at the base. And I then just simply used amounts of cadmium, copper, zinc, cobalt, nickel, and lead, and I then modeled a response of biological receptors. And we can see classic dose response for the top, except for cadmium, but in the lower one, I can actually see a lot of responsive binding. And that's because although the metal's there, it's encapsulated in the soil matrix such that it doesn't release and actually cause harm to plants or to indeed humans once ingested. And we've done a lot of modeling where we take many soils, different doses, and we can characterize how buffering and how partitioning of elements behaves in the soil. And in fact, one of the things I did in conjunction with the James Hutton Institute was then to use this across, in this case, the Kingdom of Fife, and try to understand how the different soils and the tapestry of soils across a region had different behaviors to metal assimilations. And I think it's quite an interesting overview. You can see that for zinc, there's much less binding than, for example, lead. And that's because pH and texture define the binding capacity. And what we've done over time is to then apply these to develop models that allow us to put numbers measured in the field and to use all the capacity of data we've established to enable us to be protective in the cleanup concentrations we adopt. So that's a really big breakthrough, and that was brought in by legislation way back in 2000. So I've brought in the idea of receptors, the need to protect receptors, humans, ecological, water. I've brought in the background of the types of contaminants. I think I'll move on now to actually give you case study examples of remediation and how cost effective these are. Well, it's one of my favorite slides because if you look at the literature and you look at companies that do remediation, you'll realize there's literally thousands of different methods on the market. But I just want to hone in on really what actually the differences are. So if I go to the red square in the middle here, it says remediation strategy. And I might want to remediate water rather than soil, because the water might be running through the soil and picking up contaminants. So to remediate water, I can use managing the water phase, or I can manage the non-aqueous phase, so if it's an oil that floats on top of the water, or I can have something completely passive, and I'll show passive examples. Low cost, absorption, it simply strips the contaminants out of the water. If I move into soil examples, it's a bit more complex. So the first one is passive technologies. So these simply work in the soil and complex up materials in a semi-natural way. But I can also think about removing the contaminants, which is what I usually want to do. And I can do that firstly by making the contaminant move from the water or the soil phase into a different phase. It could be a water phase where I'm washing. It could be a detergent that helps to wash it. It could be moving it into an organic phase in the soil, or it could be dealing with a non-aqueous phase. And then I've got the other method, which is how to degrade it. And I can degrade it physically, chemically, or biologically. And I'll give examples of each of these. And then finally, instead of removing the metal or the element or the hydrocarbon, I can add something that simply immobilizes it. It complexes it and stops it going any further. In fact, when we think about biological degradation, and I'm going to come back to hydrocarbon degradation, it's part of a natural process. And we know, for example, that white rot fungi will actively move into forests, and they'll take these enormous lignin molecules, which are often randomly associated, and using simple peroxidases, they'll oxidize the carbon bonds. But more importantly, once they begin to break up the molecules, we see a depolymerization process, so the molecules become much smaller. And then what we find is that these become attacked really by bacteria. So we can't think of a single magic bug to break everything down. What we actually have is processing carried out by various organisms. Okay, so case study. So I want to go to Glasgow to start with, to chromite ore processing residue. And Glasgow was host to the world's largest producer of chromium for almost 100 years, White's Chemical Works. And it was estimated that they produced about 1 million tons of waste that was then spread across the city, mainly in the East End. And here in 1950 is J&J White's Chemical plant beside Kilmarnock. You see the enormous slag piles around the site. And in fact, today, it was the area that had hosted the Commonwealth Games, and it's part of the legacy venture that's been passed on to the city of Glasgow. Now, chromium's biogeochemistry is quite complicated, but I want to pick on two key features here. The first is that you can see it's a chromium-3, which is the dominant environmental form. Chromium-3+ is not particularly noxious. At high concentrations, it won't do you much good. But in contrast, that is chromium-6+. And chromium-6+ is highly oxidized. It is present at very high pH values, and it is known to be a carcinogen with inhalation. And it's also reckoned to be a carcinogen through drinking as well. But the key thing here is that it's very difficult to turn 6 into 3. And it's quite difficult to turn 3 into 6. But what happens in Glasgow is that the processing ore produced massive amounts of 6. And the legacy of that has been really significant. But there's been a lot of different techniques around to look at remediation. This one here is a product specifically sold to try and immobilize the green minerals we can see in the sample. But I became very involved in a thing called calcium polysulfide. So it's basically, it's a calcium carbonate, remove the carbonate, and then you push in, under pressure, large amounts of amorphous sulfide, and it creates a liquid that smells like rotten cabbage or rotten eggs, but it's very effective at the reductive process within chromium-6+. And in fact, here's some work I initially established over a very long period of time, about 2 years. And I looked at samples from Glasgow, and I added different doses of calcium polysulfide. What I could then do was measure leachability, and I could see that the no treatment was up over 500, but the highest dose I used was at about 3 or 4% of that 500. So we could get an angle as to what concentration worked. Now, when we think about contaminated environments, we often draw diagrams to simplify it. And what we can see here is that the chromate ore was piles and piles in the matrix. And then we had culverted streams, which again, that meant they were sealed off, so they weren't going to pick up that material. But the key receptor was the River Clyde. And there was evidence that chromium was moving from the piles into the Clyde. So in the lab, we optimized it, we played for months on how to do it, and then we applied the system for real. And this is the remediation taking place. We can see that the boxes here, the IBCs, they're all full of calcium polysulfide. And what then happened is they were manually injected across the site at 2.5 meter intervals, down to depth of maybe between 4 and 6.5 meters. So again, you can see the 10 hosting in the background, all of these the pumps that we used to set this up. Okay, so we used about 300 tons of calcium polysulfide to treat 10 hectares of land. And what we proved was all the green areas here, the leaching was dramatically reduced. The red was a concern, but the yellow and ambers were quite positive as well. And what we've done since then is to monitor the performance over time. It's not the only chromium site I've done. This was a factory in Dudley where they plated guns for ammunition for the army. And in essence, chromium plating had been used. But to make things more exciting, this site wasn't just with chromium. It had acid tanks, it had nickel tanks, it even had cyanide, which is quite widely used to clean metals before adding finishing to it. And what we found here was a massive source of chromium across the site. And again, the pH value varied a lot. So we had pH values as low as 3 and as high as 12. But the chromium concentrations total were about 20,000 mg per kilogram. And of that, about 1,000 or so was actually hexavalent. So again, there we used a metal complexing agent, not calcium polysulfide. And you can see from this site picture here, the large circular hole was where old tanks had leaked and actually caused the sample, caused the chromium to migrate. So big scale, not quite the 10 hectares of Glasgow, but something going on for that. Well, in the abstract, I said that there was probably 250,000 orphan sites, so sites that are contaminated but no one owns across the UK. One of the areas I've realised is thousands of sites that are actually former mine sites where there's been metalliferous mining, and that's got a major impact in terms of water quality. Now, with another colleague, about 10, 15 years ago, we developed a remediation technique using whiskey byproduct material, and we called it a device for remediation and attenuation of multiple pollutants, or DRAM. And it's a low energy passive process that is able to work with solvents, but also a wide range of elements as well. And we looked at this in terms of how it performed, and we found there was very effective and positive responses. And additionally, I had used calcium polysulfide to try and look at zinc. So the idea here was to bring these two techniques together. So the field trial here is in Killip. So that's in the Northern Pennines, just south of Newcastle. This is the old mine area here. And we developed a container-based system. So the container-based system allowed us to reasonably work with thousands of litres a day, but also to have an ability to stop if anything was going wrong. So very simply, the container gets put on site. We then put a big header tank in to pick up the water from the mine. We then split the flow. One part goes through this passive treatment of DRAM, so the whiskey byproduct. And the other goes through a calcium polysulfide pulse treatment. So it's injecting small amounts every minute and then allowing settling thereafter. And then what we did with the Environment Agency was to agree discharge criteria for the process. And what we found very simply was that the calcium polysulfide removed about 82% of dissolved zinc, and DRAM removes about 94% of dissolved zinc. And zinc's a highly soluble element. So this is a really positive breakthrough, and it's been looked at for scale-up to actually deal with the process longer term, because the zinc is impacting Northumbria's wastewater. Now, close to home in Aberdeen site here, typically I don't have metals or hydrocarbons, I have both. And in this case, it's not a chicken factory, it's a chicken processing factory. So in addition to elemental and hydrocarbons, we also had a problem with animal waste as well. So large tanks of putrefying material. And again, putting in context, here's winter when remediation has been done. We're getting the site invaded by scavengers to pick up metals. But what we're also finding on the site are things like underground tanks that were never noted before. And this here is a petrol tank dating back to the 1950s. And again, hydrocarbon degradation being the key process. But today, the site has genuine value, and it's got real estate upon it, residential houses, in a criteria that met all the issues I've talked about so far. So human protection being the primary drive. Again, an Aberdeen site is a railway yard development. This is a picture from the 1970s. Train spotters can notice a wonderful 37 Delta 8 class there, and a range of other engines, locomotives. But what's important here is you can see by the sheen along the tracks that there's a large amount of lubrication oil spill, but also fuel oil spill as well. Now, we treated that by excavating the whole site, tens of thousands of tons, and then remediating it by a pile similar to what we use with the Bobcat. So a geotextile membrane, an addict to catch the water, and then covering it in a membrane, but turning it on a frequent basis. And you can see that's before, and again afterwards, we can see that genuine valuable real estate being realized from what was previously valueless land in people's mind. And then one of the things I've done for a long time has been work on a site here in northern Germany. This was a former ICI site where they made Dulux paints. And you see the TFs on this map here represent tank farms. And this is just where large amounts of benzene, toluene, ethyl benzene, and xylene were stored, which were used for the paint manufacturing. And again, when you look at the site, this is just a representation of key contaminant zones. We can see large amounts of very deep benzene migration down to tens of meters. And again, more systematic on the map. Now, of course, the problem is these are hydrocarbons that preferentially degrade under highly aerobic conditions. So the problem I had here was trying to pump air with depth down into the soil to then allow degraders to break down the hydrocarbon and produce volatiles and then capture that. So one of the issues is there's a finite amount of oxygen that will dissolve in water. It's about 8 mg per litre. So I need to contain that amount and sustain it. And I need to also make sure I've got a zone where I can pump oxygen down. That's what we did. And it's been done for about 5 years. And then we would capture the volatiles at the surface. So again, really bringing together an important venture to create sustainable remediation. And finally, a refinery site here in the north of England beside a canal. So we've got the refinery site, behind that with a lake for firekeeping. And here what we did was we basically land farmed it. So we created a concrete basin, a geotextile membrane, adding in the soil, and then at an incredibly intense scale, fertilizing it, adding other soil to act as inoculum, and then hoeing it, furrowing it, and then eventually backfilling it to the site when the issue had been left. Now, although that's sort of a run over some examples, showing that we can look at sustainable options, what I've done over the past 15 years is develop cost models to try and understand how much does it cost for that technique with that scenario to actually be remediated. I just want to give a few examples in terms of cost and then maybe think about issues for the future. So let's think of an imaginary site, and let's think about it being 1 hectare, so that's 10,000 square meters. Let's assume the contamination is just 2 meters, although some of the examples I've chosen, the depth is to tens of meters. But let's say 2 meters. And let's assume that the soil bulk density is about 1 gram per cubic centimetre. So that means that 1 hectare of soil would be about 20,000 tonnes. So that's an estimate. Again, the bulk density is quite high here. So think back to my example where I had a very low input biopile. So that was a Bobcat one, or indeed the one that was a chicken factory, because that was low input. It was turned quite occasionally. To put this into perspective, to turn 1 tonne of soil costs about £1. So if you're turning it 30 times, that's £30. So low input, low turn, it would cost about £600,000 for a hectare to be cleaned up. If I then double the rate of turning, or I'm doing 30 turns, that would push up to 1.2 million. And remember the figure at the start, I could manage a situation to make things happen quicker. If I have to do something like the ICI German site, and I'm pumping in air, it's about £50 per tonne. So it's about 1 million tonnes per hectare. But remember, that was 10 metres deep. So we probably want to put up about 5 million per hectare. The Glasgow example of calcium polysulfide, 2 metres would equate to about 1.1 million. It was down to about 4 metres. So it's probably looking at twice that value. And then the Dudley example, where I added an immobilizing agent once I'd excavated, that's about 1.6 million. Wow, I mean, these are big sums of money, and only realized if the value of the land can offset remediation. And one of the things at the moment is we're just doing nothing. So we've got all this land, which people are trying to guess will be worth money in the future, but we could be going ahead and try and get natural processes to be done with really quite minimal engineering, with something in the region of probably 50 pence per tonne. And that would allow a longer term sustainability option. In fact, over the years, I've done a variety of things. So I set up a company called Remediation back in 2000 with colleagues, and that was about environmental innovation and technologies using microbial sensors. And then 2007-8, and myself and Lee Cassidy established a company using DRAM, and that won a UK prize for business. But we need to train scientists in entrepreneurship. We need to think outside that box. But we also need to engage with scientists to upscale processes and make it compatible with engineering. The other thing is we've been listening to COP26 for 10 days. We've talked about sequestering carbon. Wow, we've got all this urban and blighted land that has the potential to capture carbon, and we're not doing anything about it. We're just letting it be barren and just letting perennial weeds come in. And far, far too much of our urban environment is grossly blighted, and we're not using it effectively. And I used to always say that remediation and mitigation of contaminated land was all about the money, but tonight it's not about the money, it's all about the carbon. We have a massive real estate in the UK, 250,000 sites, which all have the ability to be converted into carbon sequestration that often are not particularly owned by anybody. And we've got technology. This here is the scanning electron microscope we've got on campus across the geosciences, working with John Still. And I can now go beyond just risk assessing concentration. I can look at the main figure here with different elements, and I can see exactly where lead is in that sample. And I can then begin to inject emollients that might be sustainably developed to begin to combat these metal impacts. And I can connect that to using sensors to understand the efficacy of the process. So we're at a crucial point now. We have technologies that we can implement quicker now than we've ever done before. And we also have the capacity to understand, collect, manage, interpret, and use big data like we've never done before. And we can also deploy microdevices across fields to capture moisture, temperature, carbon sequestration, respiration, heavy metal concentrations, biodegradation of hydrocarbons. We can do that with devices that are remarkably cheap. And remember, by actual monitoring is one step towards remediation. And finally, for years, cost-benefit analysis for land was about net gain for developers, for privateers. But you know what? Net gain has to be about understanding the role of carbon, and more importantly, the influence that processes we perform have on the climate crisis. We need to join our thinking. We shouldn't be using lots and lots of turning with diesel engines and diesel motors. We shouldn't be adding expensive reagents. We shouldn't be trying to develop areas if they're so contaminated. We should think that perhaps the use of resistant tree species in these areas, the use of indigenous microbial populations for turning over material, that's the way we have to think forward. And I use this example because someone who's been committed to the contaminated land sector for over 2 decades, it's now the case that we have a key component to offer in terms of the sustainable fight against the climate crisis. And just to wrap up with, I want to thank a large number of colleagues from the University of Aberdeen. I want to thank Lee Cassidy for her work with me on DRAM, and then colleagues who generated and worked with me on that remedials data. And with that, I'm going to stop sharing and bring that to a close. So thank you very much for your attention this evening.
That was really interesting, Graham. Thanks very much for that. If anybody's got any questions and like put them into the chat box or the Q&A box, that would be really good.
Yeah, what about nuclear sites? So it's interesting. I mean, nuclear is regulated quite differently. So if you think about it, most nuclear problems are either unusual isotopes, and there's been a lot of work from a group in Manchester looking at some of these exotic molecules in Sellafield. I've never dealt, well, sorry, the only nuclear site I've dealt with was at the Atomic Weapons Establishment at Aldermaston, and I was dealing with specifically solvents that were there. The interesting thing about nuclear sites is that the elements we can find from power stations are complicated to measure, but also their behaviour is quite unusual. And what tends to happen at the moment is that complex agents are used to try and remove them, things like EDTA, and the like. But again, different regulator, a quite interesting challenge. And I think as we move through time, they'll become probably more important. I also think that the lessons today where we're trying to develop small nuclear reactors, we'll have to make sure that the proper containment's in place. The only other thing I want to add to that is that the policy that controls contaminated land was part of the 1990 Environment Protection Act, and it came into act in April 2000. So that actually means that it's illegal for people to now contaminate land, which means the world, in fact, means the UK should become a cleaner place and that we shouldn't have an increase in land. And I think it's interesting that as we move that way, we should see a better world. And I think, 10, 15 years ago, when there was a lot more money for development, we were moving very, very quickly to diminish those sites. So it's something to think about. Do I know of any companies that are remediating land as part of their offset program? I don't offhand, but what tends to happen is that environmental consultancies who are active in the contaminated land sector tend to also be doing wider environmental work. So a lot of the majors in this field, who will be environmental engineering companies, will be working on reducing carbon footprints anyway. But the problem you have is that the real value of offset is the owners of land benefiting from that. So previously, companies have tried to sell land to improve the balance sheet. Now I think what should happen is large landowners should be trying to offset their balance sheet by trying to mitigate carbon on their land. So I think joined up thinking is missing. So batteries should be managed through an effective waste stream process, and that goes for single-use batteries, and it will go for the high-charge batteries for cars and everything else. So it's illegal to be dumping that stuff in soils in the UK. And of course, what we've done traditionally is send that stuff to Asia or Africa for it to be dealt with elsewhere. I think as a consumer society, we need to really put an understanding of life cycle assessment and circular economy in everything we do. And I think a lot of the batteries that we've adopted in the 70s and 80s are completely unacceptable. I think we have to look at alternative features. The other thing I want to put in that context is that we've always been happy up until the past 20 years to see soil as the ultimate dumping ground. So we brought in a Clean Water Act because the Thames stank and impacted in the House of Parliament. And we resolved that by producing lots of sludge and sewage that we dumped onto land. We then had the Clean Air Act in the 50s because the cities were so blighted. We reduced burning, but what we did was we often encapsulated a lot of the waste, not just the soot, but often creating gypsum where we reacted with calcium with the sulfurous waste, and we dumped that on land. And in fact, one of the big problems we had up until early 1970s, it was quite legal to dump and make your own landfill site in your back garden, and that legislation only came in the late 70s. So we've always had this policy of if in doubt, dump it in soil. I think what we need to do is realise that has to stop. And while soil is more buffered than air or water, soil doesn't move. You can flush air away, and you can flush water away. You can try and start again. But once you've deteriorated the soil to such an extent, there is no going back. And that's why we have to be quite clever and energetic as I move forward. So it's an interesting one because, of course, microplastics are a massive problem in the sea. We know that they're in agricultural land because we've had plastic used to avoid frost damage, but also plastic used to tie bales and everything else. So microplastics are pretty ubiquitous within our food chain system. I would like to think that microplastics will not migrate directly from soil into plants, but that's yet to be seen. The damage is there. I think what we need to do is think carefully about how we use plastics, and that's been done by penalising farmers, for example, to stop using plastics so much, to charge a lot more for their waste. So I think the issue here is microplastics are ubiquitous. We just need to try and understand what sort of harm they pose. And up until now, they do seem quite benign, but there's obvious evidence now that there's associated metabolites or co-contaminants that might be of concern. And also the plastic itself can act as a absorbing centre for some nasty contaminants that could then enter the food chain. So the jury's out, there's data to be collected, that's something to think about for the future. And it's very difficult to measure plastic in soil because, you know, if I talk about lead, I can say there's the safe threshold, the risk threshold, 450 milligrams per kilogram. If I take plastic, how do I quantify that? And it's not ubiquitously spread, and it soars to the areas a lot, and the plastics are all different. So it's a much more difficult thing. But we did some work with the James Hutton a couple of years ago on one of their sewage sludge trials, and the sewage was being added to land before, you know, nowadays it's incinerated. But there was the presence of plastics in every treatment we saw. The thing we couldn't see was the migration of the plastic into the plant. And I think that's where the big interest really lies.